What is confocal microscopy? Confocal microscopy, otherwise known as confocal laser scanning microscopy, is an optical imaging technique that increases optical resolution and contrast of a micrograph. It does this by adding a special pinhole placed at the confocal plane of the lens, which eliminates out-of-focus light. Confocal microscopy also allows the reconstruction of three-dimensional structures from the obtained images by collecting sets of images at different depths within a thick object. This is a process known as optical sectioning. The confocal microscope is a special type of fluorescent microscope. A significant difference between the confocal and fluorescent microscopes is the presence of a pinhole aperture at the confocal position. Lasers, which are usually linearly polarized, are used because they are intense, coherent, monochromatic sources of light and are thus capable of being expanded to fill an aperture or focus to a spot. The laser is first focused by the excitation pinhole on only a small part of the sample, but before the laser reaches the sample, it must first pass through the excitation and barrier filter that can be either a short, long, or bandpass filter. Its purpose is to only allow the required excitation light to pass to the specimen. The dichroic mirror is positioned at an angle of 45 degrees and reflect down the excitation light to the specimen, and then allow only the longer fluorescent light to pass through it to the detector. If fluorophores are present in the sample, they emit light, which is then filtered by the dichroic mirror into the final filter. The second pinhole position in the focal plane selects only the light coming up the targeted point of the sample. The objective thus collects only this light. The surface of this sample is then scanned by moving the light beam. This allows it to construct a 2D image at a given height. Successive slices make up a Z-stack. One can then move vertically to obtain images at different heights. A volume image of the sample can then be reconstructed with proper software. Confocal microscopes allow us to detect fluorescent molecules in 3D with a good spatial resolution. Photomultiplier tubes, PMTs, are the most common detectors in point scanning confocal microscopes. An incoming photon liberates an electron from the photocathode, which starts a chain reaction so that the cascade of electrons finally reaches the anode. The main reason the PMTs have been used in confocal microscopy is because they are very sensitive. The following images display the differences between specimens for 3D imaging techniques at varied thicknesses. Thin samples do not show huge difference between confocal and wide-field microscopy. Intermediate thicknesses show significant improvement of image quality and specimen detail in confocal microscopy as compared to wide-field microscopy. The thick sample images show significant improvement in confocal microscopy versus wide-field images. It can be observed that with increasing thicknesses, fluorescent microscopy loses its clarity and resolution in the images it produces, but this is not the case for confocal microscopy. This shows that confocal microscopy is useful in the observation of specimens with varying thicknesses and can still produce high-resolution images even with thick specimens. Advantages of confocal microscopy include minimized blurring of specimens, increase in signal-to-noise ratio, increase in effective resolution, and the possibility of high-resolution microscopy through thick and highly scattered samples. An added advantage is that there are more color possibilities in images produced, since the images are detected by a computer rather than a human eye, making it possible to detect more color differences. In most applications, fluorochromes have overlapping emission spectra. Hence, the emission signals cannot be separated completely into different detection channels, resulting in crosstalk. However, if fluorochromes have distinct excitation spectra, the fluorochromes can be excited sequentially using one excitation wavelength at a time, and this is only possible with confocal systems that offer the multi-tracking feature. Confocal microscopy also allows for the optical sectioning of an object without physical contact, and more importantly, the three-dimensional reconstruction of specimens is achievable only through the use of confocal microscopy. The resolution is highly improved when using confocal microscopy. However, some things to note when using confocal microscopy is that the signal strength may be reduced by the requirement for a detector pinhole. And since this technique is more labor-intensive, users may require more training and experience to be successful in this technique. Confocal microscopy has several applications. One of these applications is co-localization. Co-localization refers to the observation of the spatial interlap between two or more different fluorescent labels, each having a separate emission wavelength, to see if the different targets are located in the same area of the cell or very near to one another. Another application of confocal microscopy is FRET, or fluorescence resonance energy transfer. FRET FRET is the non-radioactive transfer of photon energy from an excited fluorophore, the donor, to another fluorophore, the acceptor, both of which are located within close proximity, which is 1 to 10 nanometers. Using FRET, one can resolve the relative proximity of molecules beyond the optical limit of a light microscope to reveal 1. The molecular interactions between two protein partners, 2. The structural changes within one molecule, for example enzymatic activity or DNA-RNA conformation, and 3. Ion concentrations using special FRET tools like CFP, YFP chameleon. Basic operation of confocal microscopy. Switch on the machine and ensure that lasers are switched on and ready for use. Step 1. Focus sample using fluorescent microscope. Step 2. Turn the filter to empty. Step 3. Turn off shutter for fluorescent. Step 4. Click on iPod. Step 5. Click on Release Interlock. Step 6. Start scan. Step 7. Click Capture.